I'm sincerely grateful to be here this evening. It's been a long day, so the first lecture started at 10:30. We had a great time at the Swiss where we talked for several hours more with follow-up questions. And then I spent the day with Rain brainstorming potential solutions for various challenges I'm having related to contracting, ideas, anything where I can work with industry and academia to partner better to facilitate our military mission. What I'm really going to try to accomplish today is a lot. So I'm warning you. You didn't realize that you just went to the water park, but you're about to get wet from a fire hose. That's the way I present. You can ask David. He's got to see a few of mine now. I don't know how to go easy. So I'm going to really lay it on. I have a lot of slides that I want to cover, but I don't want to talk a lot about each slide that I'm presenting. It's more for you to visually look at, take it in. I want to talk about themes and show you a little bit about how we do product development in the Army. And for this, malaria is really the example that I'm using. It's something I'm incredibly passionate about. I was introduced to the subject matter about 12 years ago. That's the duration of my service. I've been educated outside of the Army at college and grad school and a postdoc before joining the Army at 30. So I had a direct commission as a captain, and I've been working my way up since then. My focus upon joining the Army was to join Walter Reed Army Institute of Research, which I'll talk about, and their experimental therapeutics division, which focuses on drugs for malaria, leishmania, and other parasitic diseases. So that's when I really first became exposed to malaria, and it bit me, and for 12 years I've been involved in this research. It's brought me to Asia, Africa, where I lived for 4 years, running clinical trial centers and performing malaria drug resistance studies and surveillance studies, to here now. And I didn't think I'd actually be involved in malaria research here at Madigan and Joint Base Lewis McChord, but I am. It happens that this is an epicenter for infectious disease research and public health. So it's just through serendipity that I'm back involved in malaria, and I appreciate the opportunity, David, to be here and talk a little bit about the efforts that I've done. This is not going to be a heavy data presentation. So there is, sorry, David, I disappointed him earlier. This is more from a program level view of what we do. So what I want you to do is sit back, relax a little bit, let your eyes soften, don't fall asleep. And I want you to appreciate the clinical view that I'm providing you. I'm going to bring you from the very large Army program level down to where we're actually executing at the field research. I'm going to loop it back around to show how we can interact with the medical treatment facilities that are a part of the Army, and that can even involve local engagements. So this is the outline for the talk. I'm going to begin talking about our major command that performs research in the Army, define the threats to U.S. service members, define the research programs that they focus in on related to those threats. I'm going to focus in on malaria, vaccine drugs. I'm going to focus in on some work that I did in Kenya, as well as how this fits into the clinical environment, which is regulated by the Clinical and Translational Research Program Office. How does that relate to the stuff I do at Madigan, which is on Joint Base Lewis McChord, just down the road? And then I'm going to bring it together by illustrating one company that we're collaborating with called Malaria that's in Seattle. So what I've done is put these place marks through. So don't be frightened by the slide number. There are a lot of place marks because I want you to think that these are the rings to the tree. Okay, so we're starting on the outside and we're going to keep moving in and in and in down this cone. So this is the major command that's in charge of doing research. This is their vision and mission. The idea here is to create products for the warfighter to protect and sustain their fighting force. This isn't always done in the academic and industrial markets. So this is a niche environment potentially where hopefully there might be spillage into the commercial market for products. The idea here is readiness to save the lives of military members. So the whole focus here is on keeping them in the fight, making sure they're ready to fight and then keeping them in the fight, whether their job is administration to being a tanker. It doesn't matter. It's full spectrum health of our soldiers and other beneficiaries. That includes retirees and family members. So it's not just about the soldier, but that's the emphasis. Why do we have to do it? Well, I already alluded to that. Sometimes there are issues that R&D and industry and academia are not prepared or equipped to deal with, and that we are the best able to serve what we need. Additionally, they may not be under time and money constraints that we just can't deal with. So we need a product to help us. If I need a new kind of tourniquet that's going to save lives, I can't necessarily wait for innovation to take place to provide it. I might have to catalyze that process to get the product that we need. Sometimes Congress says, you need to do this now. So remember when some of the up-armored vehicles weren't so up-armored a few years ago? That's an example where we had to act now, fix it, keep our soldiers safe in Iraq and Afghanistan. It is an incredibly complex process. Don't stare too hard at this. You could go cross-eyed. The idea here is very simple, that we have a full spectrum lifecycle management within the MRMC command that allows for innovation through DoD acquisition processes through the FDA. All of the vaccines, drugs, and diagnostics that are created in a way that meet FDA guidelines. There is no experimental products that are made that don't meet normal consumer regulations. As a matter of fact, there's more regulations through doing the process through the Army, because we don't want there to look like there are issues in terms of product safety and the welfare of our soldiers who are utilizing those. Some of these are very specific products that mainly the military uses. So think vaccines for various infectious diseases that most people aren't going to see, but a military member may. This is truly a global enterprise in research. So I know it's a little difficult to see, but trust me, the red words represent the Army, and those in blue, the Navy. And they're really brothers and sisters in this research product development. The red ones and the blue ones really form a network in product development and biosurveillance that covers the entire world. We need coverage of the world because wherever the threat is in the world, we have to be prepared to understand what that threat is and have products ready to support our warfighters so when they go there, they're prepared. We don't want to go there and then run into problems. I'm going to give you an extreme example of what happens to, even when we're prepared, we didn't follow our protocols. It really can compromise the mission. This enterprise is huge, not only in its locations, but its budget. It's an approximately $2.3 billion enterprise with various colors of money that support its operation. And some of those are listed here. I don't want to get caught up in them, but some of them are congressional, to Army-specific, to the NIH, to you name it. There can be nonprofit money that's coming in from Bill and Melinda Gates, for example. It's a full spectrum of money. These are the specific threats to service member health and performance that this medical command focuses in on. They include endemic disease threats, and parasitic diseases, particularly malaria, is really going to be the focus of the rest of the talk today. Chemical and biological, David is very familiar with this. Combat injuries, environmental hazards, system hazards, and battle sequelae. Each one of these actually has their own research institute within the Army to study these, and they're called centers of excellence. That ensures that they have the proper center of gravity and expertise at any one location to provide real product development. What I'm going to talk to you about today is the DoD's product development line for malaria. And really that starts at RARE. RARE is in Silver Spring, Maryland, and its focus is on infectious disease. It is the center of excellence for infectious disease. The 5 areas of focus are seen on the right-hand side of the slide. This is its location here. It's around DC, so it sits just north of DC. It has collaborating sites at multiple locations, and they're listed here, including Europe, Thailand, Kenya. Again, I served in DC at the main hub and in Kenya. I've done work in Thailand, as well as other countries throughout the world. The work that I'm going to be focusing in on today, malaria, really involves infectious disease prevention, diagnostics, vector control, and blood screening, although I'm really going to be focusing in on infectious disease prevention and diagnostics. So let's dig a little deeper. There is one program that's responsible for all of the malaria products within the Army. I can't say DOD because the Navy has a similar program, and they work together on different aspects of the problem. The goal of the program is to develop FDA-approved vaccines, drugs, and diagnostics that support the warfighter. The idea is not to get to malaria in the first place, but if you do, we can provide treatment as well if commercially available drugs don't work. Additionally, we want to make sure that we have clinical testing in endemic areas. That's why we have the overseas labs. So you can't just have an American pipeline. We have to have places where the disease is endemic. Malaria has not been present in the US since the 1950s, where it was actually part of the culture and fabric of the US. Over the last century, we really stamped it out slowly through environmental measures that reduced, the standing water, which was a big part of why the mosquitoes were present that transmit to malaria. We've had an incredibly rich history, and I entertained a lot of questions related to this today at lunch about the impact of malaria on the military. This is half the story. The story actually goes as far back as the American Revolution. Since the beginning of conflict on this continent, malaria has been there and has been a scourge. Its impact is what's called disease non-battle injuries. And what that is that means it's not a bullet that's taking me down. It's actually a disease that's endemic to the area that's incapacitating me so that I am unable to perform my duties. And most people are familiar with it from the world wars. It was crippling divisions in those wars to the point where you could see the quote from Douglas MacArthur on the bottom. That's how much extra manpower was needed. For every one division he had out, he had one in the hospital, getting treatment, and another one recovering in another ward. Malaria was killing the war effort. And so there's been a lot of money put into this because it's just a scourge that we have not been able to successfully deal with. And I'm going to talk about some of the reasons why and the challenges that we're suffering in this field. However, I do want to point out one thing which really, really recently characterized the need for continued DoD involvement in malaria, drug and vaccine development, as well as eradication efforts that are currently underway. And it's the event that occurred in Somalia. So we were involved in Somalia operations from 1992 to 94, and we had a bunch of Marines that deployed there to do some work. And what happened was, they were Marines. We're tough. We got it. When they deployed, they did not take their antimalarials. It was so bad that it immediately shut down their operations. They were so bedridden that they actually had to pack up the unit, put them on a plane, and send them home. Today, if I were to do that from Iraq or Afghanistan, so send them to Germany and then on to the US for treatment, I'm talking about transport costs of $1 million per soldier. So you can see that it's not cost effective to have soldiers getting ill in the battlefield. The idea is to keep them safe before they get sick and be able to treat them there and help them recover before we have to take them out of the field. Yes, we got great transport processes to get them out. We don't want to utilize them. We want to keep them in the fight, keep them going. If I'm talking about malaria, I have to talk about the life cycle a little bit. If you're not familiar with malaria, it's an intracellular parasite that affects red blood cells. Red blood cells are what carry oxygen in your body and keep you going. That's where this parasite has learned over millennia, since the human history began, to parasitize. They not only affect humans, I know that they affect many mammals, including chickens, mice, rats, different kinds of non-human primates, and primates, monkeys. So they go everywhere, but they start with a mosquito. It's the female mosquito that actually gives the bite that transmits the parasite. The male is not capable. And after the bite, the infectious particle will go to the liver, where it hides out for about 7 to 10 days. While it's there, it's dormant. You don't know that you're sick, and you're not affecting your work. What most people are familiar with is when it ruptures from the liver cells and enters the bloodstream. This is when you get the tertiary and quaternary fever cycles that many people are familiar with. Oh, wow, every 3 to 4 days, I'm getting that fever, and it's getting worse and worse and worse. My bones ache. I'm lethargic. I can't get out of bed. I'm vomiting. I need to see a doctor. And so this will go until your body gets stressed, and it's an asexual phase where they're dividing until a sexual phase occurs. The sexual phase is important because that's what the next mosquito picks up when it bites you. It then cultivates it again, and the cycle continues. So we have a human portion, we have a vector portion with the mosquito. The sexual portion is what allows it to keep being transmitted. The asexual portion is what kills you. There are multiple species of malaria. There are 5 that all affect humans, but the most dangerous one is Plasmodium Falciparum. For a person who is non-immune, such as most of us in this room, who have never had malaria, we would be dead without treatment, probably within 10 to 14 days. You can develop some immunity, but you have to be brought up in these endemic areas, and it takes quite some time to do. The most mortalities occur between 0 and 5 years old. There are many different research and development capabilities that this program has, and they're focused on 2 different areas, as I've stated over and over again: drugs and vaccines. We really want a vaccine to work, but we've been working on one for over 40 years. This last year marked a hallmark moment when, for the first time ever, a vaccine showed some efficacy on a large population. It was tested in numerous continents and showed around a 30 to 40% efficacy. It doesn't sound like a lot, but it's groundbreaking when you've only been able to reach 0. That is a lifesaver for 0 to 5-year-olds. It can also reduce their morbidity, so they're just not feeling as sick all the time. So really, drugs help prevent to some degree, or they can treat, but they're not the cure-all. We really need a robust vaccine that can last a while to provide protection. It'll not only help our soldiers who are in battle for 6 months to a year or a year and a half, but also help the communities where this is really an active scourge. It can help reduce their morbidity and mortality associated with this parasite. There's been many socioeconomic studies done that this disease is directly linked to poverty. It suppresses you. If you can't get out and do the work, if you can't cooperate, if you can't do your job, you can't better yourself or your family. So there are many reasons why we want to be involved in this strategically beyond protecting our own troops. They have numerous tools that help them. The drug development side is truly a fully functional drug company within the DoD. It's absolutely amazing. It has every component that you would find in any private industry out there that's manufacturing drugs. The vaccine development is one of the leaders for malaria vaccine modeling in the world. They have transitioned this technology onto other locations to make sure that there's not a knowledge sink, and one of them actually is here in Seattle with Cedar. Let's talk a little bit more about the malaria vaccine branch. Their goal is really to test, perform basic science, and test potential vaccines to bring them to the market. It's not cradle to grave. They need, at a certain point in the development, to work with a pharmaceutical company and pass it off. They're not as interested in the IP, meaning the patents, as you would think. They're more interested in solutions. They're interested in falciparum, which I already told you is a killer, but they're also interested in another species that I haven't talked about, Vivax. Vivax is more of a nuisance. Many people are familiar who served in the Korean War with Vivax. They would come back and go, boy, I got to keep going in and getting treatment. I just keep getting those fevers. And they would occur every 6 months to a year or longer. And that's because that stage actually hides out in the liver. And it could pop out every once in a while, and then you'd have to go back in and treat the blood stage, and then just wait until eventually the stores within your liver were exhausted. So they focus in on both. That one won't kill you, but it's a nuisance. So when you're looking at performing, again, operationally, your capacities are going to be diminished. Their annual budget is around 10 to 15 million. They have multiple colors of money again. Some are coming in from agreements with partners. MIDRIP is the intramural funding that comes from MRMC. That's the big command, so the outside ring. USAID has multiple engagements they do with foreign countries, so they want to see a vaccine get developed. So this is the community aspect of the R&D. And these are the core competencies. I know the writing's a little small, but I just want to focus you in on a couple things. In terms of the products, what they're really good at is evaluating vaccine candidates. They have something called a CHMI, Controlled Human Malaria Infection Model. And yes, it's exactly what it sounds like. They infect you with malaria. So if you're a human subject in these trials, you're going to get malaria, 100%. What you're hoping is that the drug or vaccine that they're testing you with will show some efficacy, so you don't. Okay, but the backup option is you're going to get malaria if it's not a successful product. They have all the services to support this pipeline. The idea is to form GMP products that are good for distribution. There's a lot of different scientific models that support this, and their staff are usually called in to work with the WHO as well as other groups on advisory panels, setting the standard for vaccine development. This is their overall research strategy for developing a malaria vaccine. They start with figuring out what epitopes they want, so what's going to create the immunity in the human. How are we going to do it? They optimize it. They evaluate it in animal models, so that's called preclinical testing. And eventually it goes into phase 1 and 2 trials, which are in human subjects. It's tough. Like I said, at this time, we've only had one that's really made it to the market that's shown efficacy at some level. The reason is that our immune systems are so different, even though we're humans, that the same vaccine is not showing durable immunity in the subjects being tested. And that's unusual. You know, you think with the flu vaccine, for example, they made many changes this year. It's a little different, right? You don't get to have the nasal one. You're going to get an injection, and they have some various different epitopes that they're customizing each year to make sure that you're protected and safe. Well, now we're dealing with something that has 4 species. There's no sameness in terms of the subunits that they're different enough with each species that they have to create their own. So that's why we're focusing in on falciparum. We don't have any immune correlates, okay, meaning what can I measure in my body that's telling me the vaccine works? Nothing at this time. A lot of antigenic variation, which I just mentioned, between the species. We need adjuvants, which a lot of people don't like, okay, because they rev up your immune system and there's questions over the safety. So sometimes less is better, but sometimes you need a little more to rev you up so that your body reacts appropriately. And we can't do this without partnerships. Remember, I said it's not completely cradle to grave. We get it to the phase 1 or 2, which means the initial clinical testing in humans, and it has to be passed off to a partner who's going to do the final commercialization. Hopefully they're interested because the government's going to be a major purchaser, but also now they have a world global market that they can sell the product to. Here's the challenge model. So in culture, they'll grow the sexual stages, which are described in that model. Okay, that's the transmissionable portion of the parasite. And they actually put it above a membrane that mimics human skin. Put mosquitoes underneath it, and the mosquitoes will feed on the membrane and take in the blood cells that are infected with malaria. Now the mosquitoes have malaria. And the human arm is bit. That transmits the malaria. That's actually how they're doing these clinical trials at this time. Their current pipeline is focused on multiple avenues. They're really looking at recombinant proteins, so this is individual subunits of proteins from the malaria that might rev up the immune system and provide protection. Gene-based vaccines, which the Navy is working on as well, so that's NMRC. And the one that I'm most excited about, honestly, and is going to be a connection, but a different company that I'm going to talk about at the end, is the attenuated sporozoite. So the sporozoite is the infectious particle that's transmitted from the mosquito to the human that goes to the liver and starts the whole process. And what they're actually working here with a company that's in Maryland is radiation attenuated sporozoite. So that's a whole parasite approach. That works great because I'm not having to worry about what individual protein within the malaria might elicit an immune response. I'm putting the whole thing in and letting the human body figure it out for me. But it's tough. It turns out that collecting all those sporozoites from mosquitoes is an industrial manufacturing nightmare. And we'll talk about some solutions to that at the end. This is actually what the product portfolio looks like. Just appreciate the picture. Don't worry about the words. The idea here is that it's full spectrum up until the commercialization. So they do everything from the initial discovery, identifying what subunits need to go in, optimizing the antigens, animal modeling, so that's preclinical, and into the human, where they're doing safety checks in addition to the efficacy. These align with the FDA standards, and this is the Army standards, which are based on engineering standards, actually. But that's how they align so that we can track in the Army and per the FDA regulations that we're moving a product along as it develops. Let's switch to the drugs now. The goals of the program are to develop weekly drugs. One of the biggest challenges that soldiers have is they don't take the medicine. We actually have things that work, but if you don't take it, it can't protect you. And how many people in here, you don't have to raise your hand, haven't finished their antibiotics that they were given by the doctor? Okay. It's even worse when you're in the field. I already told you if one of these guys gets sick, we have to take him or her out at a cost of up to $1 million to get them the treatment that they need. We want to avoid that. Additionally, we're looking for things that are called radical cure. So when I gave the example of the Korean War veteran who kept getting the recurrent malaria, radical cure eliminates all the phases from your body. So it's kind of a clean sweep, in a sense, using the drug so that you don't have to worry about that. That drug was not available back then. So they're working on new ones so that we can give one treatment that eradicates the parasite from the human body. This is their budget. It's approaching $20 million. And I'm very proud to have been a part of this group. Almost every antimalarial on the market to date, the Army has had their hands on in development. It's absolutely amazing. I had no idea the esteemed history and the colleagues that I was joining when I joined Experimental Therapeutics. I was with these guys for about 5 years and I'll show you where I worked on in the pipeline in a moment. This is the testing strategy. And David and I were laughing a little bit earlier because everybody tries to say they have a novel testing strategy in drug discovery. This isn't novel. It's very safe. It could be boring, but it works the FDA process, which is what's important. And it starts with developing chemicals that could be potential drugs and shifting them through various in vitro, meaning desktop assays to determine if they're valuable, determining if they have druggable characteristics, into various animal models. The FDA requires multiple, and for this we use the mouse as well as the rhesus, so a non-human primate, and finally into the human. It can take years to successfully work through this process. The average drug takes around 10 to 15 years to develop through the FDA at this time in the United States. This is an example of the drug portfolio. So you're going to see the same things, discovery, development, preclinical, and clinical, but they have different points at which they stop and merge. We have a full spectrum drug development portfolio, which we have to keep pressing forward. The reason we're involved in it is, right now it may be sexy, for example, for academia or industry to be involved in these things. It's not for the government. It's a mainstay. We have to support our troops. If academia or industry decide to stop tomorrow, we're going to be okay. That's the reason why we're involved in some of this drug and vaccine development, quite simply. So let me switch you over to Kenya. Now we're going to the tail end. So we're bringing that cone all the way down. We don't have malaria here. We have to bring these drugs and vaccines somewhere where we can test them, where people do. This is one example of where malaria is endemic. We have another lab in Bangkok, Thailand, that works throughout Southeast Asia at various sentinel sites testing antimalarials. I worked at MRU, as it's known, or Walter Reed Project. It's a 47-year cooperation that's occurred between the Kenyan and U.S. governments. There's around 700 staff there, and it includes mission elements in Kenya, Nigeria, Tanzania, Uganda, and really works on all aspects of infectious disease. This is a map of Kenya, and this was the footprint. These were the main sites that we worked. So Nairobi was the headquarters. Kericho was highland in the mountains. And right here on Lake Victoria, we had multiple clinical trial sites. And the blue sites are all sub-sites where we did work. So we had, and actually this is not even a complete map. We had about 40 or 50 sites that we were performing biosurveillance at for multiple infectious disease. This is an example of the laboratory structure. It's a tropical environment. The temperature in Nairobi could reach 100 degrees on average with around 95% humidity. So you can imagine maintaining equipment in this environment where you're working on generators most of the time was challenging. It was comprised of multiple trial centers, but the ones I want to focus in on are the ones related to malaria. And I was actually the director of both of these centers, the Malaria Diagnostic Center and the Drug Resistance Lab. Here's a brief synopsis of the Malaria Diagnostic Center. It was really focused in on diagnostics, just as the title suggests. We did microscopy training. We had a biobank for malaria blood films and samples where we could test all kinds of diagnostics. So we were often contacted by companies along with the WHO to test blood diagnostics. We ran multiple QA, QC programs for multiple nations and provided external quality assessments. For the US government, we were advisors, telling them, is it safe if they go to a hospital to get diagnosed with malaria? There were a lot of special operations folks running around in Africa, and sometimes they get sick and they can't make it back to their medic. So we had to assess, medical facilities to make sure they were equipped to diagnose and treat them if they needed it. The Drug Resistance Lab is shown here. It's a fairly modern lab. We were lucky. Most of them were not this well equipped. I think that we had one of the nicest labs in East Africa. There's in vitro culture and biobanking, so that's growing the parasite in culture and then saving it so that we can analyze it later, doing drug sensitivity screens. These screens are important because they correlate to the efficacy that might be expected in a human. So it's happening in a plate, but is that drug work in a plate relates to its action in a human. We did field molecular analyses, so this involved genotyping and clinical trials. Again, this was an early warning system to advise the Kenyan government, our US government, about what is happening, and also help RARE prioritize how they're developing their drugs. Because if they're not actually developing it to what's seen in the field, it's a worthless drug. Some of the most recent drugs that have come out have shown resistance in 6 months. Originally, 40 years ago, they were lasting 30 to 40 years. And so the parasite has this amazing genetic plasticity that allows it to escape all the stress that we put on it, whether we're trying to use vaccines or drugs. So I want to start making some connections. We're getting to the end. So I brought you through the pipeline from the big organization down to the field as an example on how we conduct malaria research. How does that affect what's happening in medical treatment facilities? The U.S. Army has multiple medical treatment facilities on multiple continents. One of them is just down the road at Joint Base Lewis McChord, Madigan. It's where I work now in the Department of Clinical Investigation. Well, we have a program, and it's called CTURPO, that actually fosters the clinical investigation programs that are within the medical treatment facilities. That's really the bridge between the clinical and the research. That's the argument that I would make to you. You've already seen their requirements, what they focus in on for research. That was the entire rest of my talk. You've seen that they're pretty well funded in the billions, and that they have many platforms to be able to do basic and translational research. What they're missing sometimes are human subjects, okay, are the populations that we actually need to study. It's great that I have a laboratory in Kenya where I can do a clinical trial. But a Kenyan's immunology is different than an American's immunology. They actually have semi-immunity. They're not the same. We may be humans, but they have been used to living in an endemic area, and their biology has adapted to malaria. None of ours, or most of ours in here, has. We would actually die when presented with that malaria. And so we have an opportunity to perform some of these studies at these medical treatment facilities. One of the reasons why I'm excited to be at Madigan is it has some unique characteristics that other medical treatment facilities don't possess. We have a very large beneficiary population. It's in the multiple hundred thousands. Conventional and unconventional troops. So this includes your normal troops and special ops who have different needs. We're a tri-service population, which is at most places now, and equal access to care. So we bring it all together in terms of the patients that we're seeing. We also have a very robust clinical investigation program with many graduate medical education programs. What does that mean? We study a lot of things. We have a lot of people who need scholarly activity and require it for their faculty and student status. So they need research. CTURPO is really the father of these clinical investigation programs. It's meant to steward, the programs and ensure that the quality is high for the research that they're conducting. There's 1,100 protocols in the system. That's a very robust system overall. It also serves as the tech transfer. So that's important because when I want to get an agreement in place to collaborate with an industrial or academic partner, they're who I go through. These are the various SIP sites. This is the one I work at. So very proud to be at JBLM. There are multiple others, though, that are fully competent on both the administrative research side and the regulatory side. Some include the one at Hawaii, multiple ones in Texas, and Fort Gordon in Georgia. So let me talk about where I am now a little bit deeper. Okay, so we're going from the SIP to the Department of Clinical Investigation, which is directly housed at Madigan. The idea here is to train investigators in the ethical conduction of research, not only to meet their scholarly research needs, but to educate them on this. We're in the Army. We're all supposed to be leaders. Look at me, I look like a leader. I'm supposed to be able to do any leadership job. What I feel privileged about working at Madigan is most of the people that end up leading a lot of these research institutions within the Army are medical professionals. I could be potentially training my next boss, my next commander. So it's been fantastic to work at Madigan, knowing that I'm influencing folks who, when I eventually become old and top out in my rank in the Army, these folks are going to keep growing upwards and have been exposed to great research that shaped their ability to lead research within the Army. How do we fit into this paradigm, research paradigm, more specifically that I've talked about? Right here, graduate medical education and military medical research. We're really the interface of those two lines of support. We don't just do research. It has to benefit the military. That can mean the beneficiaries or the active duty support member. We have a fairly robust program. But this is something I didn't expect, so I'm bringing it all together. I told you at the beginning that I was honestly surprised. I thought I'd left malaria behind when I came out here. I knew I was going to be involved in clinical research and more of a steward of a program and a mentor. About a year ago, through a collaboration with the Washington Global Health Alliance, I was introduced to a company called Malaria. They make malaria vaccines. And they wanted to collaborate with Madigan. I was thrilled. I hadn't really done a lot of work with malaria in about a year, and this is some details about them. They're a local company based in Seattle, and they have generated a genetically attenuated parasite, so it's missing 3 genes. So that basically makes the parasite not infectious. It is viable, it will invade me, it will go to my liver, but it never makes it out of the liver. That's great because they actually think that the liver is the most critical phase to making a vaccine. There's something mystical that we don't fully understand about the liver that actually induces immunity in the human. And so the company that I previously worked with to some degree in Maryland is irradiating their infectious particles. These guys have used molecular biology to make it less viable, okay? And so what they're actually working on, which is quite unique, are culture systems for the sexual stages. So you can produce mass quantities of these because that's what's needed for the vaccine using a system such as this. And that's an example of what a sporozoite looks like. That's the infectious particle that gets transmitted from the mosquito, crawls through my skin, into my vessels, into my liver. They're also undergoing multiple phase 1 trials at this time using the CHMI system that I described. So people in Seattle are being infected with malaria. Don't tell people. What we're interested in doing is actually assisting them in trying to bring phase 1 and phase 3 operations to Madigan. We're working on a research protocol at this time, and if we were able to do this with the assistance of Joint Base Lewis McChord's leadership, as well as Madigan's leadership and regional leadership in the Army, because it's going to take a lot of support. This would be the largest clinical trial performed in the U.S. in history for malaria. Phase 1 would involve 40 to 60 people, and the phase 2 could involve up to 400 to 600 people. That's a lot of volunteerism and patriotism that would be needed from the Pacific Northwest to support this trial. But remember, we're at this point because it's unethical for us to test some of these things, on folks in Kenya. They're not necessarily seeing a benefit at this time from some of the stuff we're doing with the drugs, and it might just be cost ineffective. One of the issues is if we treat them and then we remove the treatment, it's unethical. So we have to make sure that whatever we're providing, we can potentially sustain and meet the ethical requirements for that region. This really brings me to challenges and opportunities. I know many of you in the audience are entrepreneurs. You're looking for ways to interact with the USG, with other partners. And I really just briefly wanted to conclude with some challenges and some opportunities. These challenges, you're going to go, oh, Lord, they're the same challenges I have. Exactly. They're fairly universal, is the truth. I have scientific challenges, okay? Many companies involved in medical product development have those in their area. Mine are related to drug resistance I already told you about. It rapidly emerges. I have no immune correlates for my vaccines. I have a complex parasite biology. I need a durable immunity. So durable means long lasting. I want to be able to shoot and forget, let the soldier go about their business. And we have mentorship issues. Just like a lot of companies today, people are moving. It's really hard to get folks who are bought into your organization, that they're happily getting paid reasonably. I'm mixing some really complicated words here because how much is enough pay, okay? But enough where they're invested in the mission and feeling that they're getting, the benefits that they need to sustain their involvement with the company. Mission execution. We're underfunded. Who here isn't if you're an entrepreneur, okay? We try to leverage monies whenever possible. Regulatory costs and hurdles. Flexible contracting vehicles. Oh God, I spent a ton of time today talking with David and brainstorming some ideas for that. And then personal losses in an anemic pipeline. Again, that goes back to mentorship. If we're losing all of our senior leaders, who's available to perform the mentorship? But I think there's some amazing opportunities, and they're all related to actually partnerships. We have to leverage our finances. My goal with almost anyone that I collaborate with is to make $1 plus $1 equal 3. And so that we're coming out with a win-win scenario for everybody. I try not to be too focused on the patents, or what I call IP, so that we're really going and executing the outcomes that we need. Occasionally, we do have patents, and we do license that technology, which Malaria is actually using licensed technology from Rare. But as long as there's an alignment with the military requirements, it's pretty easy to establish a collaboration. In this case, it's malaria elimination and eradication efforts, which drugs and vaccines are directly related to. So anything related to the basic exploratory science through the clinical trials, it's easy for me to start collaborations with. For the Pacific Northwest, this is an amazing time to be here. The strategic focus for the military is Asia at this time. Southeast Asia is the hub of malaria drug resistance. It also is a potential vehicle for importation into the Pacific Northwest. There's a lot of commerce and personnel movement between those areas of the world. So we're also working with the state of Washington to make sure that they're aware of malaria cases and other things that are occurring. For the medical treatment facility, this enhances scholarly research. So remember, these guys are there, they're waning, they want to be involved in research. These kind of opportunities to work with Malaria, this might be their one opportunity to be involved in a clinical trial. That's great. I want physicians and other medical staff involved in these opportunities because it not only enhances their level of expertise and the service that they bring, but while Malaria is not the best example, other clinical trials, for example, in oncology or other things, actually enhance patient care because you're not having to go to another medical center to receive the newest of the new treatments if the standard treatments have failed. Additionally, for some of these folks, they need this brain candy to stay motivated. It's sometimes difficult for our physicians. They see a lot of patients every day, and we have to make sure that we're feeding them and keeping them intellectually stimulated so that they're involved, and embedded into it, and we're not losing them as well. So I sincerely appreciate this time today. I'm going to close with that. I've truly enjoyed getting integrated into Madigan Army Medical Center. The experiences that I've had in this country and others working on the malaria drug development and vaccine development pipelines have been pretty amazing. Most people don't know the comprehensive product development that occurs. This is just one disease that they're focusing in on. I could give you 20 more briefs that were just focused on separate infectious diseases, let alone all the other threats that I discussed briefly. I'm also very proud to be partnering with Malaria and other Seattle-based and I-5 corridor companies because it's a fantastic opportunity to leverage the experience and biotech capabilities that are developing along this corridor. So I'd really like to thank the RAIN and David for inviting me today, and at this time I'll take any questions you may have. Thank you.
So first thing, I'd like to apologize the size of the screen. I will take responsibility for that, but we'll be sending out the slideshow so you can get some of the detailed information. Some excellent slides on there. Yeah.
I told you.
Questions, come on. I've got a couple of questions. You made a comment about the drug resistance. The new drugs are, and I thought you said something that was measured in months. Is that right?
Yes.
How many?
6.
So you could go through the process of preclinical and...
Well, that was them failing in 6 months after they've been put out.
I understand. So all the effort and all the energy goes in, and a new thing comes out, and then it's introduced into the field, and within 6 months,
Yes.
there's resistance to the new thing.
Yes, that has been a case study for that one particular drug on how not to do your product development. That's not the standard, so let me be clear. I was giving a very extreme example of what can occur when you don't have your QA, QC, and linking what you're doing in a white coat laboratory, okay, in an academic environment with what's happening in the field. Your second question, sir? Oh. Hold on one second.
Somebody else can go.
Sir?
With the evolution of next-gen sequencing techniques and the advancement in CRISPR technology, has there been any attempts at actually looking at the mechanism by which the parasite binds to the liver and then trying to find the specific gene and altering that just so it's not viable?
Binding to the liver is a contentious topic by many laboratories. There are a lot of folks that have different hypotheses on how it's working. Have they applied CRISPR technology, so gene editing, okay, to that? Not at this time. Are they applying gene editing to malaria? Yes. They're particularly applying it to the interactions that are occurring with the vector, so the mosquito, so that they can actually stop the reproduction of the malaria in the mosquito so it never gets transmitted to the human in the first place. So if we block transmission and we treat humans that have it, the reservoir should dry up, is the hypothesis. Sure.
Just on the vector issue as well, has there been, so what is the, what allows the mosquito not to suffer any effects of harboring that?
It does.
Oh, it does?
It does affect its fitness. It can affect it in various ways in the literature, but it can infect its ability to survive in its endogenous environment. It can affect its reproductive capabilities. There is a cost and burden to carrying this parasite, not just in the human, but actually in the mosquito. Yes, sir.
You mentioned early on the taking away the water to eliminate the breeding grounds.
Sure.
I recall growing up in the Midwest, dancing behind the fogger that would drive around
Mhm.
the neighborhood with the spray. I was curious if, do you get into the educational side with communities that you go into to help on the prevention?
Not as much, but we might interact with the governmental agencies in the host nation that does. A lot of times the host nations are more receptive when it's their own folks that are leading the effort. So we try to work with the other Ministry of Health representatives in foreign countries to assist them. I think where we're best at actually are what I call mill-to-mill engagements, other militaries. So our military with their military. Sometimes there can be conflicts when you have mill to civ. It depends on the host nation. So civ meaning civilian. And that's where USAID and the U.S. embassies come in and actually provide up civilian to civilian interactions. We tend to do more mill to mill. Being, almost all militaries have a oligarchic, hierarchical, structure, and they tend to communicate better with one another and can allow institution of some of the best practices that might help in eliminating the infectious disease threat. Yes, ma'am.
I've heard some stories about people developing transgenic mosquitoes that would
Yeah,
also stop malaria.
you got it.
Where is that in the pipeline?
So they're already ready to field for some diseases. We talked about this a little bit earlier with the students when we were discussing some stuff at the Swiss. But recently I read an article even that there was a private company that is ready to distribute genetically altered mosquitoes in the Keys, so the Florida Keys, to really fight dengue. And the community doesn't want it to occur. The health department and others are fighting ferociously for the dissemination of these vectors. But the community is scared. It's different. You always have these mutant nightmares of how are we affecting the ecosystem, and the ecology surrounding us. I think that the science is pretty solid behind it, which is why the company has been able to produce a product that's been vetted for use. So we'll see if it's going to be deployed in the U.S. Yes, sir.
In regards to, you referenced the actual parasite itself as a very large amount of genetic plasticity. Does that hamper molecular diagnostics?
It's very hard. The parasite is actually, if we're talking about nucleotides that make up the DNA, AT-rich. And being AT-rich makes it incredibly difficult to dissect apart and run in various molecular assays. However, the newer techniques that involve next-gen sequencing and other things, they've found workarounds to get it. It's not the best, but it works. We're getting there. By the time I was leaving the lab in Africa, we were at a point where we were doing what I call omic studies, so genomic and other platforms to look at whole genome. One of the issues is when we're looking at genetic markers of resistances, you only know what you know. Sometimes you bump into them by accident, or you have a hypothesis that leads you to it. But a lot of times with our these parasitic genomes that we're analyzing, we didn't even know that things were important that are. And so by analyzing the whole genome and not allowing our mind, to limit the possibilities, we're actually finding correlations that we didn't know existed. And it's allowing us to prevent drug resistance from emerging quickly, because this can be invited into the drug pipeline early, using samples coming from the field, so that you avert that 6-month dead drug disaster that I was describing earlier. Yes, sir.
Is malaria a disease that you can see or forecast out a certain amount of time? Is this something we'll ever eradicate, or like polio, or is it something that will just always be with us?
That's $1 million question. I think that what the answer is, we want elimination. However, until we have all the tools at a capable, deployable state, I don't think that we're going to reach that goal. It's incredibly challenging, which is some of the architecture of some of the countries where this disease is most prevalent, to affect those systems in a way that will require multinational collaboration. Okay, think about Kenya. So Kenya is right here, Ethiopia is above it, Uganda's here, Tanzania's here, and trucks are driving all throughout it. If I get malaria in one place, I stop overnight in multiple places through 4 countries. I've just been a carrier for that disease. So you're talking about multinational collaboration. You're talking about environmental changes, such as getting rid of standing water, the use of bed nets, possibly augmenting or affecting the mosquito, so the vector, affecting the human biology through the use of drugs and vaccines. Treatments that include vector control, so not only drying up the water, but dispersing chemicals in the water that can kill some of the vector. Fogging. This is the layers of complexity that come into dealing with a scourge like this. And until we can wrap our hands around it in a totally comprehensive, programmatic way, it's going to be very hard to get to that end state. Yes, ma'am.
I have 2 but very elementary level questions here. So, number one, I can't imagine the compensation that someone that voluntarily allows themselves or steps up to be infected with anything. So I'm curious about that and the profile of that. And then also, is it voluntary in the military to take vaccines, or is it required, or does it depend on the...
It's not voluntary.
So these guys elected not to take the medicine prior to...
So in that example where they deployed and they failed to take their medicine, they elected not to. That's what we call a command discipline issue. And in that case, the field doctor and other folks that were supporting that unit were probably spoken to quite firmly about the need to keep the soldiers healthy so they could perform their duty. In terms of looking at the CHMI, so the controlled human malaria infections, I won't discuss the numbers specifically, but what I would say is that they're compensated for the number of blood draws. and also the number of vaccines that you're giving through a ethically approved research protocol that went through an institutional review board. And that board determines the risk to the human subject as well as the relevance of the science and marrying them together. And so they get compensated quite a bit for the risk that they're taking on. There's been no deaths associated with the trials, and they've been, Walter Reed Army Institute of Research was the first to do these trials. Actually, the individual that led the program infected himself. What they do is we have very well characterized parasites that we use, so that if the drug, the experimental drug or vaccine fails, you are given a rescue drug. And you're constantly checked so that as soon as you start feeling, even before you feel symptoms, hopefully we're catching it with the diagnostic so that we can quickly give you the treatment, because that means you failed. It's not bad, that's the science. So what I would say is that these are iterative models. So iterative means repetitive. And every time I'm going through this iterative model, I'm looking for improvements in the science. So I applaud these volunteers that do this. I'm hoping that through education and some amount of social voluntarism into this science, that we, if we get to the point where we're doing these trials at the large scale that we're talking about, that we have the voluntarism needed to be successful. So, yes, sir.
Is there any effort to eradicate mosquito as a vector, or?
That is a very interesting question, and I don't know that I would want to do that. They're an essential part of our ecosystem. It's admirable. I think what we'd rather do is augment the vector so that it doesn't carry the malaria, or the malaria is not an issue. The mosquitoes really feed a lot of things. Think about that. They're a necessary component of the environment. So I think that we want to try to maintain them. I don't want to get into global politics of global warming, but for whatever reason, the Earth is getting warmer. As it's getting warmer, the vectors are actually spreading their areas throughout the countries. So what are they bringing with them? All of these infectious diseases. And mosquitoes, in particular the Anopheles, is able to carry multiple infectious diseases. So this is worrisome from a public health perspective, because we need the mosquito, for the architecture of our ecosystem. But at the same time, it's bringing in these threats that can affect public health. So how do we manage, that in a way that's not detrimental to us as humans or the ecosystem that the mosquito is supporting? Yes, ma'am.
So you mentioned that animals also get malaria. So are those the same mosquito species and the same virus species that are?
No, so the parasites aren't exactly the same. They're different species. So I can't take a chicken one, for example, and put it in me and get malaria. I might get a little sick if I do, but it's not going to grow and develop and go through the cycle I told you about. It would probably get in me, be sick itself, and die, because I'm a different biology. And so what we're suspecting when you look back at the genetics is that since the origin of man, malaria has been an issue. It's basically grown up with mammals, and it started out as actually an extracellular component. So it's a part of a family known as protists, and actually merged into an intracellular parasite through its evolution. So it didn't start inside the red blood cells or wanting to invade them. It made its way there because it was advantageous evolutionary for it to do it. It's a parasite because we're not gaining from its presence. It's draining us. So there's nothing synergistic about, it being in us. It's not a good thing. Are we good on time, David, or should we? Because I could stay here all night.
Yeah, so I encourage you guys to go up to Jake afterwards. We'll probably break down everything. We'll probably migrate over to 7Cs and get a little nightcap, if anybody's interested. I want to thank Jake again.